So hi everyone. So I just wanted first to apologize because I was not here today morning because I would have loved to see the talks and presentations during the day, but unfortunately I was on my way here, and I wanted also to say that I did not repeat. So I did not look at my side. So I will no I look at my side. No, but I should have at least tried to see how to say. So when they asked me, I was contacted by the Student Council to do this presentation. I asked what do you want to talk about. They say, yeah, I don't know what you want. I say what you want, and I hope that maybe it's for students to know how a career for bioinformatics look like. So I'm bioinformatician 27 years. So it's very long time. So I was here not at the beginning, not of course not, but so after this is an overview of my training. So I have a master's degree in applied mathematics. I had it first, and then I did a PhD in comparative economics. And I was so lucky when I started my PhD. I started in September 1997, and my PhD started with the genome sequences. Here, genome sequences. This is was the beginning of genomics. Yeah, I was doing it before genomics because we didn't have microarrays. You don't know what is microarrays. Already, you don't know what are microarrays. So that time we had pieces of mRNA. So this was used to measure the expression of genes, and we didn't have genomes. And we tried to approach genes. This is look ages ago. So I was lucky because I was at the beginning of everything. And with the in mathematics, I was starting this is I learned together. So because when you are studying mathematics, you do it. So I learned biology for my PhD. I was working with because we had at the time. When I ended my PhD, I started a postdoctoral fellowship at the Max Institute for Molecular Genetics in Berlin. I was in the department of genomics. And when I arrived in 2001, we had a huge genome coming. So I was lucky to be able and before that we had cognitive disease, which is the youth genome coming, and silicon, which was and this I had it during my PhD, so I was introduced a little bit to UCAD, and then when I came to the maximum, the human genome arrived, and I was responsible of annotating the chromosome 21 genome. So this is lots of lab. I had to learn as a mathematician, really, because Nikki said that you learn biology in books. Of course, I have to say that you can learn mathematics in books. It's very complex. But biology you can learn still now, after 7 years, I'm still learning biology because some things I did not meet. So.
There's no sound from the
La la la. Asma? No? So I'm still learning biology, but I can say that as a mathematician by training, I can move to computer science easily. This is because I did some programming during my undergraduate student studies, and then I can move easily to data science. So when data science era came, for me, it was easy. And I had around me people who studied with me that already did data science, and they said we could develop some data science easily, and I could understand them, and I could explain to them what the biologists need, because I understood what biologists need. So it's a luck. So to be able, today morning, when I came, you have to understand what is that. When I came, I arrived, my flight landed at 11. And we took a new airline here. I took a room close to the site. And my colleagues took my luggage and I came here. I didn't know where it is. So I asked, where is the door? To me, there is the door. It was really close by the parking place where and then I just came in to the hotel. I was seeing David. He was walking. I asked him, do you know what is your meeting room? He said, no, I don't know. I wouldn't have met David. I wouldn't have seen this way. It would have been complex. So you need luck. You need to be there and to understand that you need luck and to understand opportunity, to see the opportunity coming, to know how to go forward. So before going, because I know I like to make plans. I don't like to go like this somewhere. So before going, and then I did in Berlin, in Germany. So France and Germany do Scotland do that work the same as Africa. So before coming back to Africa, I wanted to know how to proceed because I knew that I had to start a group of bioinformatics. So I met many people, I discussed it with many people, what can I do to start a group of bioinformatics? And bioinformatics in my institute. So I really talked, I was in Berlin, so I have been to the sysadmin, to the researcher, and then I have been in my way back from Berlin. I went to Marseille, so I have been to the laboratory where I did my PhD. I asked them, what are your recommendations? And here is the recommendation I found. The foundation and community. It's very crucial. The first thing you need to think about the community. First, you have to love your community. It's love of love. You need to love this community. And when you love the community gives you back. It's very important. Love is everything is turned around love. And you need the community to grow. So you give her and the community will give you. The first thing you do is give. And then one day they will give you will have that. So I was taught to be inclusive, be welcoming, and create supportive and collaborative environment. Develop a local and international network. This is what first. To focus on development and capacity building. So I asked, I am a bioinformatician. I learn bioinformatician. I should not learn people what is my science, how to do it. Yeah, they told me, but you learn them, they will understand what you expect from them. Don't be afraid. When you learn them, teach them how works informatics, they will understand what you expect from them. And when they will collaborate, they will come to you. This is very important issue. So train massively your local community and regularly organize training and workshops. Then you have to establish a leadership structure, even if it is informal. Then seek funding and resources. Consider in-kind support. Accept in-kind support. It's very important, the in-kind support, because it helps a lot. Be independent, especially in terms of infrastructure. So do not rely on institute. Even in Max Planck, the lab of bioinformatics has his own infrastructure. There is an infrastructure for the whole institute, but the lab of bioinformatics has his own infrastructure. And keep the momentum. I was told this by many people. So do not accept it. Accept from the people the same way other behave. And before maybe month before I came back to students, it was in December 2024. I met someone from South Africa who came to visit Max Planck, and it was Winston. I don't know if you want Winston Hyde. And Winston Hyde, he's the one who told me maintain this momentum, keep the momentum. Don't forget to keep. And he first of all, he laughed when I told him I'm going to Tunisia. I told him, so, but he helped me to establish this plan. So this is what I did. So I did lots of trainings of people. I managed to look for funding for research. And all these recommended. I managed to train PhD students myself, and 3 PhD students are currently being trained. I did train many, but because I believe that I would not like to have students around me. I just like to have 1 or 2, it's enough. If I have 1 or 2, and I'm able to train them very well, and at the top of research, that's fine. For me, that's fine. So I had, this is my career almost. So until now, and believe me, and the recommendation people made me really what this is. I followed all the recommendations. I might make them, of course, and so you need to have a vision when you start your career. You need to know where you want to go and what you want to do. So, and it works. So I had some other luck because we had mouse and human genome. When I came to ISCB-Africa of Tunis, we had genome. I was there in December 2024. Genome was in July 2005. So Freshman genome was there 6 months or 9 months after, and everybody in my institute worked on Leishmania. So this Leishmania genome was crucial for everything. And this Leishmania genome enabled me to apply for grants and to get funded. So we had a re-entry grant from TDR, disease research from TDR funding. We had a new grant which was Cisco, which was about already when I came back to the institute, I do system biology because it was in that period everybody wants to do system biology. So one of the first, and I had collaborations in the Master Institute that we are doing some system biology. So I wanted to continue on that direction. But so we had this funding. One of my first grants was a Cisco project. It was a Cisco biology project where we tried to model the immune response to Leishmania. And then I had this life drug, life shield. So I had plenty of funding for Leishmania. So this gave me a hand time support from my institute. They gave me a small platform, not big platform, but small platform that enabled me to work. Small servers, computers, workstations, and 10 computers. Now they're in one computer. Before we had computers that we had to put in parallel. So I had my cluster of 10 computers. So that was enough to help me for the rest. So then in 2006, I was contacted by Olivier Gasquel. Olivier Gasquel is here. He's from France, who invited me together with other 10 scientists from all over Africa doing bioinformatics to Jobim. It's a conference of bioinformatics French conference in French language, which was organized in Lyon in 2006. And following this conference, Olivier Gasquel came to me and told me, and to Dan Masiga, and told us to organize the first conference of bioinformatics in Africa. So we organized it together. He had funding from the Ministry of Foreign Affairs, French Ministry of Foreign Affairs, to fund this conference. And we organized the first conference of bioinformatics in Africa, which was at ICP. It was not the first Pan-African conference of bioinformatics. There were plenty of other conferences. But it was a Pan-African conference of bioinformatics. It was in Nairobi in 2007. And during this conference, I met all these people. I met Nikki, I met Aman, which was one of my postdocs, Dan Masika, Olivier, of course, he was there, Usama, who was student. I met Ezekiel, who is from Nigeria. I met Scott. I think Foury Joubert also was there. I met Jean-Baka. I met Apollinaire. So all these people were during this conference, and this conference went fantastic. And it was for us, we were really to be that good. And this conference gave awareness about bioinformatics in Africa, in fact. And we started the series of ISCB-Africa ASBCB conference in bioinformatics, which were organized so far in Dar es Salaam, Bamako, Entebbe, Cape Town, Casablanca, Kumasi, and we had an online session. So this is, I think, 7th or 8th conference series of ISCB-Africa ASBCB conferences. And following this meeting also, we had another meeting which was in Nigeria, and we were invited to write a grant on to help Africa develop their network of bioinformatics sort of. We had the first proposal, and which was not successful, but our second proposal was successful. And we had funding, which was ISCB-Africa, the Pan-African Informatics Network, and which lasted 5 years, which is great opportunity that made African bioinformatics my family, because I met these people every year, and I discussed with them, and many projects came out of this conference. And this conference also helped me a lot in my institute locally, because we had 2 important tools that were given by this conference. There is the IBT training, Introduction to Bioinformatics training, which was great, and which we had organized in the institute every year. And then we have extended outside of the institute. So we expanded from the organizing IBT at Institute of Tunis. So every year we had 20 students coming from different laboratories from the institute who had attended this school. And between 2 years, we didn't have the same students. And some of the trainees became trainers. So they came to this training to help. It was sort of train the trainers when they did a good, they showed good understanding of the training, some trainers. So this helped us to have trainers also. And we had this node assessment exercise. And in my team, I told them, and even we sometimes included people from other teams when necessary. I told them, you have to do it. And they did it. So sometimes you say, you have to do it, they do it. It's easy. So this was really, these 2 things, these 2 tools for me were fantastic to help me in building this network in my institute and the capacity in the institute. Amazing tools. And then during SAOBNet, I did something fantastic. It was, I don't know, maybe some luck also. I was co-chair of the AfSCS Harmonization Working Group. And during this working group, we talked about harmonizing the phenotypes across different. And then we went to standardizing the page report forms that were translating these phenotypes. And from that, we developed a tool which standard some CRF for all these, covering all these, how can I say, all these thematics, for example, how to measure alcohol, tobacco, and other substances, anthropometry, cancer, cardiovascular, and also during this phenotype harmonization, we started, which is African population ontology. My colleague Malik Shoush will talk about it later during this conference. So this was, for me, it was not really bioinformatics, but as a mathematician, by training, I have seen that there is an importance there, that we can compare only comparable things. And if the genomics is different and the serum are different, how can we compare? So for me, it was very important to be able to these serums, to ask this question the same way, in order to be able to compare the genomics. So this is what we have done, and this serum is available for everyone and can be downloaded by everyone. Well, we go back to the institute now. So this was done for ISCB-Africa. We did many other things, but these are some important things, I would say. So bioinformatics is sitting in between biology, medicine, computer science, math, and physics. So at the institute, so it's during my first period in the institute, I was focusing on comparative economics. We need sequence analysis. We need to understand sequence analysis. We organized plenty of trainings to enable them to acquire some genomic data analysis skills, knowledge about major bioinformatics algorithms like neighbor joining or cluster. Develop statistical skills, learn how to use Python or Linux. Programming skills are like Python per shell. Maybe you don't know about per anymore, but it was develop training to develop databases, and like the latest was to develop analysis skills. So people want to do DNA data analysis, then they wanted to do RNA then shutdown, then 16 ads. So each time, and we had to think how to help them to acquire this knowledge. And this period of training, I think it lasted this. So from 2004, December 2004 until 2005, 2007. We organized plenty of training. But in the meantime, I was training students also, because you need to have a group. We had here an informal group, but to have a formal group, we needed to move to a formal group. We need 10 students, 50 students. So we at the beginning to have the colleagues around the institute to train a team by informatics. Some of bioinformatics. Also, we needed to train 50 students by informatics. So in 2006, we had enough manpower to be able to apply for a laboratory and to become a formal laboratory. So this is what happened. And this laboratory is still there until now. So what is this laboratory? The laboratory we created is laboratory of bioinformatics, biomathematics, and biostatistics. So we have seen over the last year, we have seen that we had different groups. In fact, we had people that were bioinformatics, so I was bioinformatics. We had people who were doing bioinformatics, which is they were doing and people who were doing biostatistics. And in 2016, we wrote this project of laboratory, and we had this team, and each team had a program, a work program, and the Ministry of High Education and Research asked us every 4 years to update our work program and update our team. And within the laboratory of bioinformatics, biomathematics, and biostatistics, have decided that there will be a turnover in the top of the laboratory. So the head of the laboratory will not stay the same all the time. So I was the first head of the laboratory for the first four years. And then my colleague Simen bin Minad, who is biomathematician, was head of the laboratory. And then Aysar Dira, who is bioinformatician, now is heading this laboratory. And he will be heading 2028, and that time we will decide who is going to be the next head of the laboratory. Maybe it will be me, we don't know. We will decide when the term of 2008, at the end of 2008. But to keep the equilibrium, to keep, because I have how to develop this bioinformatics team. So to keep my strategy or everyone's strategy, I'm head of this bioinformatics team since the beginning. So the head of the laboratory, team leaders, they cannot to get some logic or okay. So this laboratory was in the institute. We had 9 research laboratories. The team of bioinformatics was collaborating with a 9 research laboratory, was developing research. He was helping the different laboratories to develop or to implement their research. So we had were using public data or non-public data, and we had IPT collaborators or non-IPT collaborators in this framework. So here at this point, I was able to develop, and thanks to the training, because all the young researchers I know me in the institute and they come to me, I was able to develop international thanks to AfSCS network, and in the institute, thanks to the training organized by us or by ACA-BANET, we were able to implement and to have an institutional network. Both are very important. You cannot have international. We cannot have institutional network without having international, and you cannot have international without having institutional, because you are there to your institute to do research, help people in your institute to go forward. So what are the research objectives of the bioinformatics team at BIMS? So biological questions are addressed as systems. So me as I said, when I left the institute, so when I came to the institute, I was focusing on training people and looking for funding and having an infrastructure. And I was not able to really work on system biology. But what I was trying to do at least was to address the biological question as a system, not to look at one genomics or transcriptomics alone or proteomics. The cell works as a system. So this is the way we were approaching this the biological question. So we were implementing approaches were able to analyze, integrate systems into the system, any know-how and any omics data, metadata, clinical or social data. We were studying infectious diseases, especially leishmaniasis, but also non-communicable diseases such as diabetes or Crohn's disease or sickle cell. Then came, I don't remember when, but came the idea of precision medicine or personalized medicine. So when it came, I have seen that it was very important. So it was around 2012, something like this, or 13. I have seen that it was very important to start thinking about precision medicine and how to implement precision medicine in a very difficult effort, because in North Africa, I don't know how it is in Sub-Saharan Africa. I think it's South Africa. We don't have a reference, we don't have reference variants. We don't have anything. And the pharmacogenomics data which is available on public databases is developed on Caucasian, Europeans or Americans mostly, or maybe some others, but there was nothing for the North African population. So we have to start developing. We have started discussing about the precision medicine to develop awareness and to see how we can organize to, at the end, with a very low cost, try to apply precision medicine in Tunisia. So we had lots of discussion. There is someone very important in that area with Yusuf Hamdi. He's really pushing precision medicine. So we discussed it together different aspects, and in order to start this foundation for biology. We had the good foundation for biology, genetics, and computer science. And for us, it was important to be able to translate it into precision medicine. So this I did it before being able to start working on system biology. What about system biology? System biology is integrate different variants, integrates various fields of study, including genomics, proteomics, metabolomics. Its objective is to construct comprehensive predictive models and simulate the behavior of biological systems under various conditions. What does it mean? So we have, once we have proteomics, transcriptomics, genomics, or even genomics, we want to understand what is because for example, you have sometimes for some genetical diseases, twins, identical twins, they do not respond the same way they do not develop the same way the disease. And this comes from the cellular functioning, which is different from one individual. They have genetically very close, very close genetic background as they are identical twins. But they don't eat the same thing. They don't eat the same thing, which means they don't have the same metabolomic. And metabolic, when you change the metabolomic, this means that you change probably the proteomic and transcriptomic. So the idea was to take the cell and to see using data, transcriptomic or metabolomic and proteomic, and to see how this impacts on the cell behavior. So while reading about, because you need to do a lot of research, while reading about system biology, I have seen that there was Cobra, which is a oriented framework designed to meet computational science associated with system biology. So, as I said, I was interested on doing system biology since 2005, since I left the Max Planck Institute. We wrote this Cisco project. We were able to do some networking, understand how the cell works, but we didn't really do system biology as I wanted to do it. So following that, I made the first attempt, which was around 2013, with first project with colleagues from Nigeria who came to the institute to pastor, and we started using Cobra to try to model immune response to Leishmania infection. So it was around 2013, after 6 or 1 month or 1 year, wanted to go to the US. He decided that he doesn't want to do any PhD. So he did some job, but it was not final. So it stayed the way he left it. And then I took a postdoc because even though I was very, very busy doing many other stuff, I still wanted to do this system biology thing. I took a postdoc, who did one exam and she didn't manage to do it. So, and I didn't have energy to focus because you need lots of reading. You need to focus a lot to be able to do this system biology thing. So lately, in 2014, 34, I had a grant, which is International Agency of Atomic Energy, where I was supposed to do some modeling of the immune response of to vaccination. So I started working on doing this pathway using Cobra Pi for CBAS. It worked. And then in the meantime, I had Feriel is here. Feriel, who started her master, and Feriel gave her the project of modeling the immune response to infection. And it worked. We worked together, and it worked for both of us. And we used the Cobra Pi, which is an open source platform that was dedicated to running system biology. So for the European CBAS, how did it work? So we took a zebrafish genome scale metabolic model. So this was done. It was published here. There is a reference. It's not visible, but it's a reference. It's a product. You have mathematics where you have genes, and metabolites, and you have the reactions. So you have the matrix here. The lines will be genes or metabolites, and the columns will be the reactions in which these genes or metabolites are involved. And this way, you see how the reactions encoded in this. When you have expression data, you can overlap this expression data with your metabolic model to see within that condition how the system behaves. And you can do some what is called balance analysis. You can decide some genes to decide which genes are not anymore or are existing anymore in the matrix. And there is that expression behind the Cobra and behind this matrix. And there is a tool that enables you to test the system if it is equilibrium or not. So the flux balance analysis is sort of you have the system that when you take out some genes or proteins, it will show you how when the system equilibrium the equilibrium be reached in the system, and how will the system look like at that point. So this is what zebrafish, we had this zebrafish model, and there was a CBAS genome in ensemble. So we used orthology to map the zebrafish genes to the CBAS gene, and we obtained a model. So there is a poster if you want to see it. We obtained a model, and now we are currently trying we are analyzing RNA-seq data and proteomic data to see how RNA-seq data of infected CBAS, so it will be CBAS infected with nodavirus. So this RNA-seq data to see how the response of CBAS to the infection by nodavirus, and we had a proteomic data, which is the response of CBAS to vaccination. And we had also this metabolic network modeling of Leishmania infection that was done by Feriel, and she will present it. And there it's magic because you can take. We did it on mouse macrophages. So we took mouse metabolic model, and we took Leishmania metabolic model, and there is a technique that enables and then to do any kind of experiment on it. And what we did is we did at the dual RNA-seq data. We found the dual RNA-seq data of mouses infected by Leishmania. So we could, in the combined model, we could measure the expression during the infection and obtain a host pathogen first, a host pathogen metabolic model, where we coupled both models, and then when we added the expression data, we obtained a Leishmania host infected macrophage model, and we ran the flux balance analysis, and which was amazing because this project is much more in-person, of course. Periel is a student, so she's putting all her energy in this model. But that student anymore. So the other model is not that advanced, but this one is very well advanced, and it's amazing. Is that in one click? It's not really in one click. With a good, when you integrate the correct data and you analyze the data properly, in one click, you could. When she ran her flux balance analysis, she ended up with results that my colleagues that are experts in Leishmania infection could validate, and all the hypotheses, work hypotheses, that were highlighted by the model obtained by Korea. So for me, it's very successful, and the paper is being written for that. So, but then, of course, we had this we had lots of be able to see properly here to be able to system to do. I wanted to do it in 2005, but in fact, I was able to do it in 2024 because I was very not focused. I was unfocused during that period. So between 2005, I couldn't focus on doing my own research. I could only focus doing my own research only when I gave back some responsibilities, and I could do it to be able to make it successful. But some of the other responsibilities you see here, next slide, is family. Family is very important, and the family has a timeline which is different from your research. So I try to take you backward. This is something that you have to integrate into your system to get things to be able to advance, because it's very important. So, and also here I wanted to, well, slide change it a little bit. I wanted to acknowledge, because during my career, I met some people were very nice. Most of the people were very nice. And you have these 10% were very good. And these 2% were so good. But most of the people, I had amazing. Most of me as women had kids. So I had most of my colleagues men, they were pushing me. And they were understanding it's very important. Hussein Daleji was my first director general. My second director general. Really, they were really pushing me and helping me. And each time I asked for something, we had helped me. I had Aida, Karim. She was the head of the laboratory where I was before doing the films laboratory. Lina Babush, really much. But one day, I thought we had very important, and we were deciding established in the institute. And there was one laboratory which was so I love the laboratory of bioinformatics, because I explained that I had enough with me to be able to do the bioinformatics. Belgium. And Lina was head of his laboratory. You shut up. If he can say that, probably the lab will not be here. Oh, I would have had problems now. But he asked, and he was, both were doctors, and he was his professor, and he said, shut up. Here is all the problem. And my postdoc, Malik, he is fantastic. Malik is here. You, Emna, Usema, I cannot. All of them were fantastic. Nabil, Ines, she's doing, Ines is amazing. Ines did learn, did numerical sort of ancestor of artificial intelligence. And she's working in a school of statistics. And she has amazing data science students. And she's bringing all of them to the lab, and they are doing projects. So, and she's bringing a huge component of data science. Nabil's mother also helped me with data science, and all of them with their recommendation, with their friendship, with. But you still have this 10%. But you can make it. Don't expect everybody to be Anthony. Everybody is not Anthony. But focus on people's issue and positive, be positive, and love people and science. Voila. How long?